The overall goal of this procedure is to identify signaling events induced in viable responder cells following their physical interaction with nearby dead cells.
This method can help answer key questions in the fields of cell biology, pathology, and physiology, such as the influence that dead or dying cells exert on their live neighbors. The main advantages of this technique are that it is straightforward and inexpensive, and that it can be applied to study biologic effects induced in live cells following their interaction with neighboring cells undergoing apoptosis. Demonstrating the technique will be Lanfei Feng, a technician in my laboratory.
To begin this procedure, after passaging the BU-MPT cells onto 100 mm vacuum gas plasma-treated tissue culture dishes, grow them in culture medium A to confluence in a humidified 5% carbon dioxide atmosphere at 37 degrees Celsius. After that, rinse the adherent cell monolayer 3 times with culture medium B. Then, induce apoptosis by incubating the cells in culture medium B containing staurosporine, a non-selective protein kinase inhibitor, for 3 hours at 37 degrees Celsius. After 3 hours, collect the medium containing floating apoptotic cells that have detached from the monolayer. Subsequently, centrifuge this medium for 10 minutes at 500 times G. Afterward, discard the supernatant, wash the pellet 3 times with culture medium B before adding it back to the adherent cells. Following this washing step, detach the remaining adherent cells by adding 5 millimolar EDTA